Babies are built out of stem cells, which are crucial to the future of the baby's immune system and future life. Our longevity and our health is based in part on how many stem cells and the quality of them that we get at birth and how long they survive in the body. There was recently a study of a 115-year-old woman, and they looked to find out, she donated her body to science, and they found that she was still living on 2 lines of stem cells to make her blood. The placenta is a masterpiece of construction, orchestrated by stem cells and churning out millions of stem cells into every crevice and even into the amniotic fluid, where those stem cells orchestrate the lung and gut integrity. In utero, the baby breathes, swallows, and urinates stem cells, as well as having them transported inside via the umbilical cord. There are about 80,000 stem cells in 2 mLs of amniotic fluid. However, at birth, the baby should receive a massive stem cell infusion from the cord blood. An immediately cord-clamped baby is deprived of its own property, which is approximately 8 million stem cells. The problem today is that many obstetric staff have lost their grasp of core physiology, so that even in 2014, hospital doctors and nurses often have never seen or done a delayed cord clamp. The function of these stem cells at birth is to dampen down the effect of the very high catecholamine stress hormones, which are actually vital to jump-starting that baby's immune system at the time of birth. The stem cells allow for a seamless transition in the baby for the next steps into life, one of which is the establishment of the gut and body microbiome, all those microbes. Profound anemia also results from immediate cord clamping. Up to 40% of that baby's blood can be left behind, and it's a contributor to much illness in infants today. Amazingly, even today, there are many in the obstetrics world who are uneducated and into thinking that a baby is in a privileged, sterile environment up until birth. This is what I was taught when I was doing obstetrics. But through DNA fingerprinting, we can tell that microbes are in the placenta, the amniotic fluid, the umbilical cord, and many other places, including meconium, which means that the baby has been exposed throughout pregnancy to microbes. What we have here is meconium samples of 4 different individuals. This is an adult for comparison. This is a baby from a healthy mother. This is a baby from a mother who had gestational diabetes. This is a baby's meconium from a mother who had type 2 diabetes. And this is just the color coding of the different microbes. And what you can see is that they're very different from person to person, depending on, actually to group, depending on the status of the mother. One of the most important elements of a healthy child is a healthy microbe profile in the mother and the baby. And that's dependent on doctors understanding what normal really is and understanding what else they can do besides napalming the gut of the mother and tossing antibiotics into a situation when they don't need to be. The key for a mother and a baby is that the baby's immune system is about to be taught how not to respond, to develop tolerance over a period of time, so that it learns in a protected environment what's safe and what's not. And again, the key to this education of that baby is the mother. The maternal microbiome changes during labor, as I just mentioned, to become anti-inflammatory. Changes during gestation to be anti-inflammatory and then becomes inflammatory at labor. And then it changes during the feeding process, as I mentioned to you. So the baby is brought into anti-inflammatory after birth, where they'll stay for 2 years with that slow transition into a slightly pro-inflammatory profile. Mother's milk continues to maintain the baby anti-inflammatory, just like the placenta was. Antibiotics, antifungals, poor diet with low minerals, low fiber will put the mother-infant dyad at a disadvantage at any point before or during this period. This schematic here shows us the different colonization times of the infant based on its age. The major task of a baby's immune system is to know when not to respond. All of our immune systems right now are constantly sampling and screening material from within our own tissues and also from the exterior. And 99.99% of the time, it has to make the decision not to mount an immune response. A well-developed anti-inflammatory immune system is dependent upon microbial balance. Which bacteria dominate during the initial colonization process is very important, as is the stepwise introduction taking place at the right times. And we can see the shifts of the different bacteria that come in later. Whether or not the colonization process happened on time and appropriately can impact allergies, inflammatory bowel disease, autoimmune disease, and brain function. This is well documented in the medical literature. There are 3 classes of bacteria. One are called commensals, which you're probably familiar with. They're residents of the bowel and the body that don't cause any harm, but they don't really help. Then there are other ones called symbionts, which are residents that actually give benefit. And then there are microbes called pathobionts. They're residents that can cause disease under certain circumstances. For instance, Clostridium bacteria are actually very beneficial. We need them. But if we throw antibiotic into the system, what happens is their competitors are decreased, and then they can overgrow, and then they become problematic. That's an example of a pathobiont. But note that all of these bacteria are residents, normal residents, and that balance is the key. This one here, Bacteroides, is a symbiont, meaning that it gives benefit, and it helps keep inflammation down. And as you can see, it's one of the first colonizers of that baby. So not only do antibiotics affect this pattern, but any kind of repetitive and constant stress or vaccination and the mode of delivery and even the mode of feeding can affect how this happens. Until relatively recently, it was assumed that microbes in human milk were contaminants from the mother's skin and that human milk was sterile. But when they collected milk under aseptic techniques, bacteria were still found. So scientists were mystified, and they decided to look at the microbial DNA fingerprint and were astonished to find that human milk bacteria had different DNA fingerprints when compared to that mother's skin bacteria. A vaginally born baby gets its first gut flora bolus transplant from its mother's vaginal flora, which is hugely complex. And this is flushed down into the gut by precolostrum, clear fluid, and then that's followed by colostrum bolus, and then ends up in mature milk, which still continues to have microbes in it. There are approximately 10,000 bacteria per ml in aseptically collected human milk. But that's only part of the story. The milk that looks like a white liquid is actually a live transplant made from the mother's own blood. Human milk is a pulsing living organism which has one main function outside of food, and that is to keep everything in the baby as much as possible anti-inflammatory. Breast milk has substances which stop pathogens attaching and other substances which go after invading pathogens and butcher them to pieces. Development of the intestines and mucosal immunity and growth of many of that infant's tissues are mediated by human milk. That master butcher that I mentioned before is called Hamlet. It stands for human alpha-lactalbumin made lethal to tumors. It kills tumors. It's being used by the oncology industry, or they're attempting to anyway, to treat cancer. But it happens to also be lethal to pneumococcus and encapsulated bacteria. Now, if you think this is a long list here of what's in milk, look at stem cells, microRNA, messenger RNA. We've got erythropoietin making blood. We've got factors that make bone and orchestrate bone. We've got food for the baby. We've got food for the microbes. There's all kinds of stuff going on there. But this is actually a really short list. Because every time scientists look with new technology, not only does the list grow longer, but they find multiple functions for many components of milk that were formerly assumed to have had single functions. And Hamlet is a really good example of a protein that can be either a food or a killer. And we used to think that proteins only had one function, but it turns out that dependent on the shape of Hamlet, it's either going to become a killer or it will be a protein food. A mother's breast milk is unique to her baby, meaning that if she has a premature baby, she's going to make milk that's good for that premature baby's brain and immune system, and it changes to suit that baby, and it even changes throughout the course of a day, believe it or not. It does way more than flex a muscle. It's pretty much a miracle in the making, yet many mothers don't know that. But how could they, when scientists are only just beginning to understand the functions? But one thing that's becoming blindingly obvious to scientists and anyone who reads breast milk medical papers is that human milk contains factors that are capable of programming the infant's developing immune system and metabolism into a health-promoting model. Yet it does way more than that. It can heal and it can rebuild. I've got papers showing that a wet nurse without a disease actually cured a genetic disease in a baby that was suckling from her. That's because of the messenger RNA and the microRNA. Trying to remember the author's name offhand. If I remember it, I'll tell you. Remember that placenta that I showed you before. Well, IRMAK, I-R-M-A-K. Scientists who understand how a placenta builds a baby, particularly when it comes to microRNA and messenger RNA, realize that breast milk performs many of the same functions as the placenta did in pregnancy. So, one big key to a baby's genetic destiny is the fact that a new mother's mammary glands take over from the placenta to provide the developmental guidance to ensure a baby's genetic destiny is fulfilled. It's setting the baby up for the perfect development. Now, this doctor who said that was the guy who developed who discovered, the guy who developed stem cells is up there. He's the guy who discovered them. So let me remind you that breast milk is 100% alive, with millions of live cells, including stem cells, microRNA, messenger RNA, cancer nukers, immune primers, bacteria, viruses, probiotics, proteins, carbohydrates, oligosaccharides, fats, and all this stuff that we just seem to think of most of the time as food. At least I used to, and most of the medical literature did as well. So the natural bias of breast milk is anti-inflammatory. You should all get that by now, as well as having key roles in epigenetic modulation and immune system education, particularly relating to tolerance, meaning that the baby's not going to react strongly to things that it shouldn't be reacting to. So let's look at how vaccines might impact the natural programming of the anti-inflammatory bias that nature has set forth for millennia. Before we do, though, we need to discuss a major flaw in scientific thinking, which is that scientists and doctors think about the newborn immune system like that. There's a concept that on one hand, a baby is so vulnerable immunologically that it needs vaccines to survive. But on the other hand, it's stronger than an ox, and according to some doctors, they can tolerate 10,000 to 100,000 vaccines all at once and will do no harm whatsoever. And there's also the idea that vaccines will make a baby stronger. It turns out that neither one of these are true. This thinking comes about partly because of flawed assumptions as to what a baby's immune system does. In reality, a baby's immune system looks like that. It's a fully developed, gleaming race car with a top-grade computer with an extra program on top of that adult program which temporarily clamps it down for a very important reason, as well as an inbuilt security blanket. What is it? There she is. When that baby meets a threat and starts to react in an inflammatory way, the breast milk is right there to kick out pathogens from inside and constantly redirect the immune system to react to the minimum degree possible. There's an article by Pabst in 1997 that shows that if you vaccinate with measles, mumps, and rubella, a breastfed baby or a formula-fed baby, their immune profiles are completely different. Okay? The breastfed baby keeps being brought back into the anti-inflammatory state. The clamps are an extra program superimposed upon the baby's normal immune system for two very specific purposes. And to do that, a baby's immune system has to be anti-inflammatory. Those two purposes are that a baby needs to develop good gut flora and be gradually exposed to food and other potential allergens via the environment and through breast milk, which is the best way for a baby to learn what's safe and what's not. Both food and other allergens are processed by the mother and presented to the baby encapsulated in breast milk constituents, which tag those things as safe. All the scientists who study breast milk and neonatal immune system in depth know that the developing immune system is highly susceptible to modification and disruption by environmental influences, and it's at significantly greater risk than the fully matured immune system of an adult. The same applies in all mammalian species. In terms of milk, modulation of the immune system. But what's important for us is that numerous unique immune maturational events are programmed into specific windows of prenatal and neonatal development. These maturational events are building blocks on what subsequent changes in the immune system are built.